So, welcome everyone to Scientists to Go. My name is Zoe. I'm one of the educators here at the Gulf of Maine Research Institute. And before we get started, I just have a couple of things to go over. This is a 45-minute program. So first, our presenter will take about 20 minutes to introduce himself and his research, and then you all will have time to ask him questions. So if you think of any questions during the presentation, feel free to put them in the chat and I'll read them out to our presenter at the end. Today, I am very excited to introduce our speaker, Dr. Nathan Furey. Nathan is an associate professor at the University of New Hampshire, where he researches the movement and food chains of different species of fish. So with that, I'll go ahead and turn it over to you.
Some fantastic. Well, it is really nice to be chatting with you all this morning. And thank you for that introduction, Zoe. Yeah, so I'm really excited to share a little bit with you all what I do. But before I get into the science that we do, I figured I would kind of let you know how did I get to the University of New Hampshire. Um, and as well as recognize that we are a really big team. So everything that I'm going to show you today takes a lot of teamwork and working together. So I'm really glad that I get to work with a variety of students and technicians and staff that kind of make all of this work possible. Now, on this map, I grew up actually in New York and kind of in a very landlocked part of the state. My closest neighbors growing up were cows on dairy farms, but ever since a little kid, I really love the ocean, and so I wanted to make my way there eventually. And so after I finished elementary school and middle school and high school, I went on to go to college, and I first went to the University of New England, and that's where I got my bachelor's degree in marine biology and environmental science. And that's when I really realized that I loved research and I loved trying to understand marine biology better. So then I went on and did a master's degree at Texas A&M. And then I actually spent 6 years doing my PhD and postdoctoral work at the University of British Columbia in Canada, in Vancouver, British Columbia. And now, for the past 7 years or so, I have been back in New England at the University of New Hampshire. So I'm very fortunate that I've been able to really work across the continent and even to a degree across the world, focused on marine animals. And I also kind of want to tell you a little bit about like what does my day to day look like as a scientist and as an associate professor. So sometimes I get to be out in the field. I actually get to go outside and look for fish and do interesting things with them that you'll learn about shortly. So this is me electrofishing with the team. So this is how we can actually sample fish in the water. And you get to look like a Ghostbuster while you do it. Sometimes I get to be out on boats. So here I am in the Gulf of Maine, working on sampling and tagging an Atlantic cod that you'll see a bit more. However, a lot of the times I'm not in the field. So sometimes I'm also in the lab and I'll have a lab coat on and looking at things under a microscope or whatnot. So for example, this is the stomach contents from an Atlantic cod. And so I can go and look at that stomach contents and try to ID things in it. So you can see which part of this is a fish, which part of this is a crab. And hopefully you can even see a couple of little lobster there on the screen as well. But other times, I am just working on a computer. So whether I'm analyzing data or I'm writing, there's a lot of time I'm also doing things behind a computer. So one of my messages for you all is that if you're interested in science, is that there's lots of ways to be involved in science. You can be out in the field, you can be in a lab wearing a lab coat, or you can be playing on a computer as well. And all of these things are really important to the work that my lab does. And I do this because I love fish. I've been very fortunate to work on a variety of species, again, across the continent, and we're going to touch on several of these today, but certainly not all of them. And so some of the big questions that I want to kind of focus on today are why do fish move? Where do they move to? And we're going to focus on the specific term called a migration, which is a specific type of movement. So first we're going to define this word, migration. And so what a migration is when you have large amounts of animals, so large numbers of animals that are all moving towards the same place at the same time. And so usually this happens in specific seasons, and they just all move together in the same direction at the same time. And sometimes this can be hundreds or even thousands of miles that they're traveling. And we see these migrations across all sorts of animals. So whether it's monarch butterflies moving from North America and the U.S. to Mexico, or whether it's animals on the Serengeti, or all of the birds that migrate across our continent every year. However, fish also undergo migrations. And these migrations can be pretty incredible because they can occur both between freshwater and the oceans, or just in the oceans by themselves. So here we've got examples of Atlantic salmon in the top left, American eel in the top right, sockeye salmon, which are a species found on the west coast in the bottom right, and then the bottom left, alewife that we have here in New England as well. And so a lot of my research is to try to understand these migrations. How quickly do they move? Where do they move to? And what makes them successful? And so the way that we track animal movements is through a technology called telemetry. And so telemetry is when we put an electronic tag on or in an organism. So kind of an example would be if we had this trout here. And sorry, there we go. My animation seemed to be a little bit slow. I apologize. You put that tag into a fish, and then we put out listening devices or listening stations. We call them receivers, and that's what that black cylinder is on the right. And we put those in the water. And if a tagged fish goes by, these tags are actually sending out sound. That's why we call them acoustic telemetry, but they're at a frequency that we can't hear, but the receivers can. So if that tagged fish goes by close enough to one of these receivers, it will actually get logged on that listening station. So we would then know when we go pick that listening station up and we download it onto our computers that, okay, this specific animal came by at this date and time. And this allows us to understand the movements of individual animals, kind of like how your phone can track your movements as you go about your daily business. And so if we put out enough of these listening stations, we can actually track animals for very long distances. So this is some of my work out on the West Coast on baby sockeye salmon, where we tagged them in a lake, and then we tracked their movements. All those little red and blue dots are individual fish that are migrating downstream, and these are fish that want to get out to the open ocean that would be on the left-hand side of the screen. And we can just kind of track these fish, and you can see some fish move faster than others, some move slower, some don't make it the entire way. So these are all things that we can look at using this telemetry technology, and there's going to be lots of examples of this in my talk today. And so, what are we tracking here in our backyard in the Gulf of Maine? Well, I'm actually going to hone in a little bit closer to home for me, which is in coastal New Hampshire. We have this estuary, so kind of waters where the freshwater and the ocean meet, called Great Bay. And we're doing quite a bit of tracking work here. And so, one of the species that we're looking at the movements are is the rainbow smelt. And I like to call them the forgotten migratory fish because there's a lot of fish in our water, fishes in our water that undergo migrations, particularly between the ocean and fresh water. But we tend to think of other fish first, like salmon and sturgeon and river herring or alewife. But rainbow smelt do these migrations too. And historically, they were actually really important fisheries. Um, so during the winter time, people would go onto the rivers and into the bay and set up these little ice shacks and cut and drill holes into the ice and go smelting. And they would catch these little rainbow smelt. And people really like them. If you fry them whole, it tends to be the recommended way to eat them. And even though a lot of people don't realize it, they really come right into our backyard. So this is downtown Exeter, New Hampshire, and this hopefully you can see the people in the middle of this river. That's my team working alongside New Hampshire Fish and Game to sample these fish because they come into our rivers to spawn every spring right as the ice comes off. So they're literally spawning and completing their life cycle right in our backyards, and they are found up and down the coast of Maine as well. And so their life cycle requires them to be in both freshwater and in saltwater. So in the winter, they come into the estuary and into the rivers. And then in the spring, they spawn in freshwater. And then they're going to, after they spawn, return to the estuary and out eventually to the coast before coming back into the estuary and into the river sometime between fall and winter. But we actually didn't have a good sense of exactly when some of these movements happen. And so that's where my lab used acoustic telemetry. And so we tagged a bunch of these rainbow smelt, and we tagged them in different rivers that are color coded. And you're going to see the little rainbow smelt bouncing across the screen, and they're going to be color coded to those rivers. And so we learned a couple of really interesting things. One is that unlike a lot of other migratory fishes, rainbow smelt will actually bounce among rivers. So a lot of fish will only go into one river, spawn there, and then leave. However, we found several rainbow smelt bouncing between, let's say, the Bellamy River and Oyster River, or between the Oyster River and the Squamscott River. And so they might actually be spawning or laying their eggs in multiple rivers. The second thing, and I'll just play this one more time if I can, is that all of these fish, there's a little time bar, it's a little hard to see at the top, but all of these fish left by about the first week of May. So that means they were done with spawning and ready to go back out to the coastal ocean at this time. And we weren't sure exactly when this timing occurred before. But this just shows you how we can use telemetry to follow the fish. But these tags can be used on much bigger fish too. So up and down the coast, there's active telemetry projects on striped bass, on sturgeon, and yes, some of you put this into the chat as your answer, also sharks. So the Atlantic White Shark Conservancy down in Massachusetts have tagged a couple hundred great white sharks over the past several years. And what's really cool about this technology is that even though my lab isn't actively tracking sharks, the receivers that we put out to track rainbow smelt and other fish can detect the tags on the white sharks. So, for example, some of our work in coastal New Hampshire and off of Portland, Maine, we have a bunch of these receivers, kind of in where these black boxes are on the little map on the right. And I'm sorry if that's hard to see. But even in just 1 year, we detected all of these white sharks. And so all of the white sharks get names. You can see how big they are. And this is all available on the Atlantic White Shark Conservancy's website. And I'm happy to share that share the link to that with folks as well if they want to see all the white sharks and where they get detected up and down the coast. And we can track movements of these animals over really large distances because of how many scientists put those listening stations out. So all of the dots on this map are a listening station. So my lab is responsible for just the handful of dots that are around the New Hampshire coast and southern Maine coastline. But there's researchers up and down the coast working together to track the migrations of these animals. And I just want to show you, oh, so just each of those dots are one of those listening stations. And so I just want to show you a couple examples of this. So we've also been working closely with Dr. Matthew Ogburn at the Smithsonian, who tracks river herring leaving Chesapeake Bay. So if anyone has family in Maryland or around Washington, D.C. or Delaware, he's tagging these fish that you see on the left all the way down there. And you might be thinking, well, why do I care about Chesapeake Bay fish when we're in Maine or New Hampshire? Well, let's see where some of these fish go. They go all the way up into the Gulf of Maine, and one of them, the red dot, went all the way up into Canada before working its way back. And then we're going to actually see some more dots pop onto the screen, and we're going to see these fish work their way all the way back to Chesapeake Bay. So this is hundreds of miles that these are relatively little fish are traveling every single year. And that animation was made by a postdoc in my lab, Dr. Mariah Livernoy. Now, another cool critter that Dr. Matthew Ogburn's lab tracks that doesn't quite come into the Gulf of Maine but travels a really long ways are cow-nose rays. So these are closely related to sharks. They have skeletons made of cartilage, just like white sharks. Um, but obviously they don't eat the same things as white sharks. But they've been tracking the movements of these cow-nose rays for about 10 years now. And I'm just going to show you hopefully it will play about a year's worth. Whoop, let's try that again. My apologies. Things are just a little slow on my end, it seems. Just give me 1 second. Here we go. And so the red dots are female cow-nose rays and the blue dots are male cow-nose rays. Now, these animals are not taking the highway down to Florida. It's just part of our animation. It doesn't force them to stay in the water. It's kind of just drawing them as they go, receiver to receiver. But it shows that they really use most of the Atlantic coast. And we'll play that one more time. Um, and they do this every single year. So they use Chesapeake Bay to kind of feed and to get ready for their migrations, but then they head all the way down to Florida before coming back. So if any of you go to Disney over Christmas or whatnot in the winter, know that cow-nose rays are kind of doing a similar thing. So I've only shown you ocean examples of this so far, but I do just want you to know that we can use this technology in freshwater as well. So we have a project in Otis, Maine. I don't know if anyone is close to the Bangor area today, but this is Floods Pond in Otis, Maine, and we're tracking Arctic char. And this is one of the very few populations of Arctic char in the lower 48. Um, and what's really cool about this work, I won't bore you with kind of the math, but in a small system like this, we can actually what's called triangulate or trilaterate the animal's position to just a few square meters. So we know exactly where this fish is every 2 to 3 minutes. And we also have depth sensors on these tags, so we know how deep the waters they're using as well. So this technology works in any type of water, whether it's freshwater, an estuary, or the ocean. Now, I do love fish, but we also track other types of animals as well, including crabs. But I want to focus today a little bit on seabirds. And so this image on the left is a common tern, and hopefully you can see, and I don't know when I bring my cursor up if you can see it. But this little black line here is a little bit of a tag. Now, because these seabirds don't really swim, we're not using acoustic telemetry, we're using what's called GPS telemetry. So these are tags that talk to satellites in space. And that's what allows us to track their movements. And what you can see is that this is an adult tern that's feeding its adorable little chick a fish. And that fish is a little herring. And so these chicks can't fly, and so they rely on their parents to bring food back for them. And that's what you're seeing a little video of on the right. And so I work closely with scientists at the Shoals Marine Laboratory, which is a series of islands just off of the coast of New Hampshire. They're about 7, 8 miles off the coast of New Hampshire, and it is a breeding colony for these common terns. And Dr. Elizabeth Craig runs this program, and I'm very fortunate to work with her. Um, but these GPS tags show us where these terns go to feed. Where do they go to get those fish to bring back and feed their chicks? And this is research being done also by my PhD student, Alia Caldwell. And so all of those lines you're seeing are terns leaving the colony, which is right here where my cursor is, and then they're going and flying around and finding food. Now, all of the blue dots you're seeing, that is where we think the terns are finding food, where they're slowing down and stopping and getting food to feed on. And so, what we learned is that these terns have certain places they like to feed. They like to feed in our estuary and in the Piscataqua River mouth. They like to feed at the Merrimack River mouth. And then they also like to feed in these waters around Cape Cod. And this is really interesting because this is actually a national marine sanctuary known as Stellwagen Bank National Marine Sanctuary, where there's a lot of sand lance, which is a fish that terns find very, very yummy. And what we find is that in certain years, there's better fish that are available, things like herring. But then in other years, um, that they're finding less ideal things to eat, things like bugs and shrimps and a fish known as the butterfish that they actually have a hard time swallowing because they're so circular in shape. Now, we generally focus on the movements of these terns during the summer when they're at the colony and they're feeding their chicks. However, these birds travel even further. So this is a bird that we tracked for an entire year. And I want you to see how quickly it moved from North America all the way down to South America. And it spent its entire Christmas and January holiday off the Amazon River mouth, which I just think is so fascinating. I've always wanted to see the Amazon River. But then come back spring, we just missed it. But over just a few days, it traveled all the way back from the Amazon up to Virginia and then all the way back up to New Hampshire, ready to lay more eggs and have more chicks. Whoops. Okay, I just have a couple of more slides because the seabirds kind of showed that they move so that they can eat or so that their chicks can eat. But in fact, for fishes too, they move so that they can eat. And we can learn a lot about a fish by what it eats. And so, for example, we have some beautiful Atlantic cod on the left. And when we look at what they eat, we see things like this and these and this. And so I'm thinking in your classrooms, you're probably already identifying what all of these things are. But this is lobster. We have a bunch of little crabs, and then we have shrimp. So these cod eat a lot of invertebrates or things that don't have spines, but they do eat some fish, particularly cunner. And so, what's common across these organisms is that these tend to be animals that you find towards the bottom of the seabed. And so, cod tend to hang out towards the bottom of the ocean. And so, it makes sense that they're feeding on things that are found in deeper waters. We also look at the diets of striped bass, too, and we see a big difference. We see that they eat a lot of squid and they eat a lot of herring. And so these are animals that are constantly swimming and are swimming up higher in the water column closer to the surface. And striped bass are a lot more active than cod. They swim a lot faster. And so it makes sense that they're able to track down much faster prey and eat things higher up in the water column. And so, what did we learn today? One, that animals move and that they can move really large distances. And this includes these really cool things called migrations, where lots of animals are moving at the same time in the same direction to the same place. And that animals are also moving to eat, and that we can learn a lot about an animal by what it eats. And so I want to thank you all for listening today. There's my email if any of the classrooms do want to reach out to me or have additional questions after this and after the Q&A period. Don't be afraid to reach out. And so I will stop there and we'll see what questions we have from folks.
Great, thank you. We already have a handful of questions in the chat, but if any classrooms have questions that come up, feel free to throw them in, and we have a good amount of time to get through all of them. So we'll start with a question that I find so fascinating as well. How do fish know what season it is? How do they know that
Yeah,
it's time
oh,
to
I
migrate?
love that question. That's a great question, right? They don't have calendars. They don't know it's December 4th today. So how do they figure this out? Well, they can sense even without knowing what day it is, right? They don't necessarily know what time is, but they know what their environment feels like. So just like how we now need to wear our coats and our gloves and our hats when we're going outside because it's getting colder as we transition from fall into winter, fish are going to feel changes in the temperature in the water that they're at, and that can induce or cause them to start moving or making changes. Oftentimes, they're following temperatures up and down the coast as they get warmer or cooler. They can also sense things such as changes in the daylight. So right now, it's getting dark much earlier than it gets dark in the summer. So they can actually track, we call that photo period in science. They can track that changing amounts of daylight as well that can cause them to realize, ooh, maybe now is the time for me to start moving or start doing something differently.
Very cool. Um, and then there was another question about the fish that are able to live in saltwater and freshwater. So it was what makes some fish capable of living in both salt and freshwater, but not others? And then does that have something to do with when, sorry, does the season at which they migrate and their biological processes for living in these different types of water depend on each other? Like, is that, are those related?
Yeah, definitely. And so we call this osmoregulation in science. How well do you regulate how salty your body is? And so when you're in saltwater, fish need to be able to keep salts from coming into their body because it would be too salty for them. And in freshwater, fish need to hold on to their salts or their electrolytes. Think of like when you eat, or sorry, when you drink Gatorade or Powerade, they need to keep those salts in them and not lose them to the water. This is done through several processes, but one of the powerhouses of this are the gills. So we think of the gills as being important for fish for the ability to breathe, be able to extract oxygen out of the water. But because those gills are up against the water, they're also really important for osmoregulation. So there are specialized types of cells in the gills called osmoregulatory cells that either kick out salts if they're trying to if they're in salt water, or if they're in fresh water, they're built a little bit differently to pull in and keep salts and prevent salts from leaving the body. Now, for animals that have to migrate between both, what we tend to see is that they have both types of those cells in their gills. Both the types that pull in salts and push out salts are in the gills, or they will change them seasonally. So if they know if it's going to be a spring migration, weeks or months before that migration occurs, they'll actually start building more of the right types of cells in their gills to be able to do that. So that's a really fantastic question. And it gets to kind of that previous question because we think there are cues going on in the environment to kind of tell that fish, hey, the season is changing. You better get ready to go travel to a different salinity of water. Start changing the cells that are in your gills to do that. Some fish, though, can any time of the year just travel between fresh and salt water. We call those fish euryhaline, and it just means they're happy in any salinity. And they just tend to always have both types of cells in their gills at all times.
Cool. Um, and we have some questions about how you are able to tag the different animals that you track.
Yeah.
So firstly, how do we tag the birds, like those satellite tags, the GPS tags?
Yeah. So there are a couple different techniques that researchers use across the globe. Um, But the technique that we use is called a harness. So they kind of get like a little backpack. It's kind of like a neoprene like material, like a thin, think of like a thin wetsuit. And it kind of goes over the shoulders of the bird, and then the tag gets attached to it on the back. So that the tag is it's attached to the back. And it's actually designed so these birds, when they're at the colony, they're eating a lot themselves. They're also feeding their chicks a lot. And it means they're kind of at their biggest or their roundest at that time. So the backpack tends to fit quite well. And then what happens is when they get ready to migrate long distances, they actually don't want a lot of extra weight because they're flying thousands of miles. So they actually lose weight, and generally the backpack then falls off. And then it's not on them anymore. And that's what made that one animation of the tern flying all the way to the Amazon so incredible, is that that was just a turn that the backpack stayed on for the entire year. And obviously the animal did fine doing that, made it all the way back to the colony the very next year. But that gave us what we think is the first high resolution full annual migration of a common tern. And we just had that paper accepted in the literature as well, describing that tern's migrations.
Wow, congratulations. It's very
Thank
cool.
you.
And then there's another question from the same school about how we're able to tag the fish, like get the trackers inside of a fish.
Yeah, so we do this a couple of different ways. So if it's a fish that's small enough for us to actually bring aboard or handle easily, usually it is a short surgery. So and all of our protocols get approved by veterinarians, so they have to know that we're doing things in a way that minimizes harm to the fish. We actually anesthetize the fish, so we actually put them to sleep for a little bit. We do a small incision, insert the tag, suture it back up, let the animal recover, make sure it's doing okay, and then it's on its way. However, something like a white shark is way too big for us to do that with. And so my colleagues, like Dr. Greg Skomall, who tags the white sharks, he actually uses an external dart. So he actually, in a boat, kind of comes up alongside the shark, and then the tag will be attached to a little line with a little bit of a dart, and he kind of gives a little stick, a little jab, kind of like us getting a shot at the doctor's office, to have that tag kind of hang out outside of the shark as it swims by. But it's actually really good for the shark because it means it minimizes how long you're holding that shark. You're not pulling that shark out of the water, which could really stress it out.
Cool. One of our questions was how many tags go out in a year? And
Yeah.
then if like how many different species you would tag.
Whew, yeah, and so it really depends on the project. Obviously, we want to tag enough animals that we can get what we call a good sample size, or it kind of increases our confidence in that, hey, this is what fish actually do. Because if we only tag one or two, maybe it's just a really unique fish, or maybe it's a fish that does something different from the rest of the population. So most of our studies, we're trying to tag at least 50 or so fish. And some of these projects we've done year after year. So, for example, for rainbow smelt, we try to put out 50 tags each year on rainbow smelt. We have a project on Atlantic cod and their movements that I didn't show you today, but we've tagged over 100 Atlantic cod with that. And some of my work on the West Coast, we deployed over 1,000 tags on little salmon. In terms of species that I have tagged, and I also see the question of, is it anything beyond birds and fish? So we have tracked crabs as well. We've given crabs little backpacks. So we've tracked green crabs that are a non-native or invasive species in our waters. We've tracked blue crabs that are native to our coast, but are generally aren't found in New Hampshire or Maine, but now are because the waters are getting warmer. We've tracked Jonah crabs that are people love to eat their claws. They're very yummy. And then just in terms of other species, we've tracked. I showed you rainbow smelt and the common terns. We've tracked Arctic char both in Maine and also in the high Arctic in Canada. We've tracked sculpin in the high Arctic, bull trout out in the west coast, sockeye salmon on the west coast, steelhead on the west coast. Um, Oh goodness, I'm forgetting others, but we've tracked a number of critters over the years, which is a lot of fun.
Yeah. There was a question about the cow nose rays and what they eat. And then also, I'm not sure if you'll know this, but we're wondering what blobfish eat too.
Oh, that's a great question. And I actually don't know what blobfish eat. So blobfish live in the deep ocean, right? And so it's very hard as scientists for us to get down there and study them. And in fact, blobfish actually don't look very blobby when they're down in the deep ocean. They look much more handsome, in my opinion, when they're under pressure. It's only when we bring them up from the deep ocean, they don't have that deep pressure on them anymore, that they tend to not look quite as attractive. But my guess, most deep water fishes are what we call generalist predators because in the deep ocean there's very little food. So that means they're going to eat anything they can fit in their mouths. So probably a variety of invertebrates and other fishes. Now, cow-nosed rays, again, they don't have big mouths with big sharp teeth like a shark. Instead, they have mouths that kind of largely point down, and that's a really good clue to us that they eat things off the bottom. And in fact, they do eat things off the bottom. They eat a lot of invertebrates and a lot of shellfish. So things like crabs and scallops and clams are all things that are eaten by cow-nose rays. And I think, in fact, I'm going to share my screen one more time, if that's okay, Zoe,
Yeah.
because I think I was ready for this question, maybe. So I want to show you this is what cow nose ray jaws look like, and this is what their teeth look like. And so rather than having big, sharp, triangular cutting teeth, they're just these flat, hard plates. And so this means they can just be really good at crushing things. And so again, we can look at an animal's mouth and its body shape and its teeth and learn a lot about them.
Wow, that was really cool. I'm so glad you have that on hand. Um, we had a couple of questions that have to do with migration.
Yeah.
So like what's the largest population of migrants that you'll see, like a big group of migrants? And then there was another question about any animals that might migrate alone.
Yeah. And so we have animals that are relatively solitary, and therefore won't migrate in as big of schools for sure. A lot of our sharks are this way, particularly our larger sharks and what we would call pelagic sharks, which just means open water sharks, sharks that don't associate with coral reefs and such. So oftentimes um, if you kind of zoomed out, there would be other sharks nearby, but they're not really traveling together, if that makes sense. But other animals can really travel in really high densities. And so a lot of my work on Pacific salmon on the West Coast, those baby salmon, for example, I worked on in a lake system where every spring, over about a 4 week period, between 10 and 70 million salmon left that one lake. And there's over 100 lakes in that region that all have sockeye salmon populations doing things at the same time. So there are times we're talking about literal billions of fish that are moving in the same general area at the same time. It doesn't mean they're all in the same school and that they're all friends, but that they're all moving across these oceans at the same time, which is pretty darn incredible to think about.
Yeah, there's a lot of fish runs that you can see even here in New England where there's just so many fish that they're flopping out of the water and going back in.
Absolutely. So in the spring, like if any of you or your parents are big striped bass fishermen, oftentimes they want to go where the menhaden are, or what other people call them pogies, or where the river herring are, because these are really big migrations along our coasts in Maine and New Hampshire, where we can have hundreds of thousands or millions of fish even. Moving along the coast, moving up into our rivers to spawn, and those striped bass are following them because they want to feed on those. And so that's very common to see just the water boiling with these fish, particularly as the striped bass are scaring them.
Cool. We have one more question about the anesthesia that you give to
Yeah.
the fish when you're tagging them. But does it have any impact on their biological processes? Does it affect them afterward?
Absolutely. Yeah, this is a great question. So, I just had to take my dog to the vet this past week where he had to be given a sedative, and he was a little slow for a while, right? And just like us, we're not quite the same when we come out of it. So, one, we try to make sure that we give them plenty of time to recover because we don't want to just release that animal and have it not be able to respond to a predator, for example. So we make sure that they're swimming really strongly, that they're responding to our touch and our presence, and we're only releasing them if they look 100%. The other thing that we do and is really common in our work is that yes, not just the anesthetic, but right, we just captured it, we pulled it aboard, we put a tag in it. These are all things that, might make you a little bit tired or might make you a little bit stressed. And so oftentimes what we do with our data is we ignore um, some of the behaviors that we see early on, and we don't include them in our analyses. We don't consider them true behaviors of the fish. They're probably an impact of us handling the fish. So, and we can sometimes even see that in our data of like, ooh, first 24 hours, this fish did not move. And then it started to move around and started to look like a normal fish. That's a really good clue to us of like, hey, let's take off the first couple days of data here if we're trying to assess what does a fish do in the wild. And that's a very common practice in our field as well, and one that we have to be thinking about constantly. We want our data to represent what these fish do in the wild, so we're constantly looking at our protocols and procedures to say how can we minimize the stress that we're putting on these animals for their sake, but also for our sake, so that we're actually learning real things about these animals that can help conservation and management as well.
Mhm. There's another question about fish, and we're wondering if they sleep.
This is a Oh, I love this question. This is a great question. Um, so it's very hard because we can't ask a fish, right? We can't ask a fish if it dreams or if it sleeps. It's very hard in a biological sense to define, are they actually sleeping or not? Now we know that a lot of fish at least rest. And we can do this through things like looking at their heart rate and respiration rates, and as well as things like brain activity. That we can see, yes, it seems that animals will, for periods of time, and kind of like us, follow a daily schedule. So if they're what we call diurnal and they're awake during the day, we'll kind of see these responses at night. Or if they're a nocturnal fish, we'll see it during the day. But it's very hard for us to define sleep because fish also don't have eyelids. So they have no way of closing their eyes when they sleep, even if they wanted to. But there's also some animals we're pretty sure if they don't sleep, they're getting very, very close to it. So one example of that is the parrotfish that some of you might have heard about because they actually feed on corals, and they have big beak-like teeth that are really kind of cool looking. But they also have another really cool adaptation in that every night they cover themselves in a blanket of mucus and they just kind of rest in a mucus cocoon and don't move for several hours. So we're pretty sure that they're either sleeping or it's very, very close to it. It's a very deep rest. Now, other animals we know can't truly sleep. For example, some sharks have to swim to breathe. They're what we call a ram ventilator. So they have to swim with their mouth slightly ajar through the water to get water to pass over their gills and to get oxygen. So if they stop swimming, they will stop breathing. So those animals have to always be swimming. So they can't be truly sleeping, but some scientists have hypothesized that they actually have some really cool adaptations neurologically or in their brains that kind of let them be on autopilot, such that they're actually kind of sleeping. They're not really thinking about things, but the body's awake enough to keep swimming. And there's sort of, this question's so great because it's an active area of research that scientists don't have the answers to. So maybe some of you will go on to be scientists and help answer this question better in the future.
Yeah, that would be really awesome. Um, Another question from the same school is what happens if a fish is passing a transponder and then dies?
Yeah, so we will continue to pick up that tag on a receiver, provided that tag is close enough to a receiver to be heard. And so oftentimes, what happens if an animal dies is that we might just see that fish stay at the same location for a long time, or that tag stay at the location for the same time. And we actually have criteria in our analyses to be like, okay, that's probably not an animal not moving. It might just be an animal that has passed. Um, some of the times, an animal that we tag might get eaten by a bigger animal, and now we're actually tracking the predator as well. And sometimes we see behaviors of like, oh, that is swimming way faster than we know that that fish can swim, or it's swimming in the wrong way of a migration. So we might be tracking a shark, or we might be tracking a seal now, and we have ways to identify that in our data as well. Now, if another animal eats that tag, obviously it's going to digest the animal that it ate, and it will eventually pass that tag out of its system, and it will fall to the sea floor. And usually within a few days, that tag will get covered up, and we'll stop hearing it, depending upon the environment and what the currents are like and what the substrates or sediments are like.
Cool. And then I'll just end it with one final question, which is, what's your favorite part about your job that you get to do?
Oh man. I have a lot of favorite parts of my job, so this will be hard. But I think anytime, at its core, my job is about learning new things. Just like you're all in classrooms there to learn about new things, my job is to learn things that nobody has ever learned before. And to me, anytime we can do that and then share it with folks, what we've learned about these very cool animals and show how cool and complex they are, to me, that's a really exciting and wonderful opportunity. The other favorite part of my job is that I also get to train a lot of people. I work with undergraduate students, I work with graduate students, I work with postdoctoral scholars, so people who already have their PhDs but want to advance their careers further. And anytime they really feel that they have learned something and they've progressed and that they've become a better scientist. Like when things click for them and they're like, oh, I get it now, I understand it, and they can go on and be strong, independent scientists themselves, that's really rewarding and I really enjoy that.
Yeah, well, thank you so much. I love this presentation. I know that a lot of our schools did as well. And thank you everyone for attending. We will be back in at the end of January with our next presenter. And I hope to see you all there.
Thank you, everybody.